So Desire is a 5-year project, which is now in its last year of activity. We will conclude our activity in October. The main purpose of Desire is studying the epileptogenic processes when they arise from developmental processes in the brain, and then study also how the epileptogenic process and the developmental process influence each other. The name is Strategies for Innovative Research to Improve Diagnosis, Prevention, and Treatment of Children with Difficult to Treat Epilepsies. And the project is balanced, is about 50% clinical and 50% basic and laboratory research. Okay. So why have we chosen to work on epileptogenic developmental disorders? Well, they are caused mainly by malformations of cortical development. Also, we have a number of conditions which are designed as a collective name of epileptic encephalopathies, which in part may be malformative, in part may be non-malformative. They are the most frequent cause of drug-resistant pediatric epilepsies, and unfortunately they carry a lifelong perspective of disability and reduced quality of life. So collectively they represent a major medical and socioeconomic burden. Not much is known about their pathogenic mechanisms. So what are the main objectives of Desire? Well, we have been studying the genetic and the epigenetic causes of the epileptogenic developmental disorders, particularly malformations of cortical development, but not only. We have tried to make our best to clarify the molecular networks and the disruptive protein complexes which are involved. And from this step, we have also emphasized the next step, which is the one of identifying new diagnostic tools, not just in terms of biomarkers, but also in terms of clinical practice in the sense of a better knowledge of the disease process. And then the next step is to work for treatment options using both randomized controlled multidisciplinary trials, but also working in experimental models. How it's the structure of Desire? Well, there are work packages which have been working in trying to identify as many as possible causative genes involved in these developmental disorders. Then, from this molecular work, developing new diagnostic tools and biomarkers, developing, according to the new findings, functional experiments in animal and cellular models, and also in human brain slices. From this step, then also trying to develop innovative therapeutic strategies and developing randomized clinical trials. So I'm almost finished. It's a 5-year project, as I mentioned. There are 28 partners, 8 third parties, 9 countries. We have calculated 2,000 and almost 300 months and more than 250 researchers. This one has not been patented by Desire. Okay, this is the list of the partners. We have no time to go and see them all, but they are all in the possibly next slide. Okay, thank you for your attention.
Okay, so now we will listen to a short presentation of EpimiRNA from David Henschel.
Thank you very much. It's my great pleasure to give a short overview of the EpimiRNA project, which is focused on microRNAs in the pathogenesis, treatment, and prevention of epilepsy. First of all, who are we? We're a group of 10 academic research teams based at universities in Europe, North America, and South America, and a group of 6 industry partners that are working to commercialize some of the research from the project. Prior to EpimiRNA starting, virtually none of us had ever worked together as a team, so this was a really new research project. And also we had quite an innovative balance within the project. We had a third of the group which was basic epilepsy research teams, mainly using in vivo models. We had a group of clinical neurologists based at epilepsy specialist centers in Europe, and then we had a third of the project which was specialists from outside the epilepsy field, experts in RNA chemistry, protein identification, and mathematical modeling of pathways. And I think this has been one of the most exciting and enjoyable aspects of this multidisciplinary project. So our project focuses on all things microRNA in epilepsy. At the start of the project, there'd been a number of breakthroughs linking these molecules to brain excitability and to epilepsy, but there were a number of important questions that were still unanswered. MicroRNAs are small, non-coding RNA molecules that function within cells to control gene expression networks. They do this by binding directly to messenger RNA, so protein coding messenger RNA, and when they bind to that messenger RNA, they bring about a reduction in protein levels. So they're the ultimate information integrators within the cell. Individual microRNAs can also target many of these protein coding messenger RNAs, so they are network targeting molecules. And perhaps what better a molecule for epilepsy, for a network disease, than a network targeting molecule. We focused on adult temporal lobe epilepsy in EpimiRNA, and we had 4 basic projects within the consortium. The first of these was to try and discover new microRNAs that regulated brain excitability and to understand how they were doing this, understand the underlying mechanism. We would explore microRNAs within biofluids as potential diagnostic biomarkers. We would look for the first time at whether or not there's genetic variation in microRNAs in patients with temporal lobe epilepsy. And finally, try and develop microRNA-targeted therapies. We had a set of 9 work packages. Work packages 1, 2, and 3 took brain tissue from animal models and epilepsy patients and looked for new microRNAs. Work packages 1 and 2 also had a biomarker component where we would extract microRNAs from fluids such as plasma and cerebrospinal fluid to look for microRNA biomarkers. We had a genetics work package. 3 work packages focused on trying to elucidate the mechanisms and targets of the microRNAs, and 2 work packages focused on microRNAs as potential therapeutics. During the course of today, the presentations will give an overview of what we've achieved so far, and of course you can't summarize it on a single slide, but I think some of the main areas are on this slide, and they include scientific progress towards really understanding what these molecules are doing and why they're capable of influencing brain excitability. We have achievements in terms of moving some of our research projects towards preclinical and clinical development. We were able to secure a patent on a therapy based on a microRNA for epilepsy. We've developed some community resources around databases for microRNA epilepsy research. And finally, some of the researchers within the project have gone on to secure funding for larger epilepsy research projects, so increasing the sort of critical mass of epilepsy teams in Europe. Thank you very much, and I hope you enjoy the EpimiRNA presentations today.
Thank you. So this is the overview of the EpiPGX project.
Good morning. I'm speaking on behalf of Sanjay Sisodia, who won't be arriving until a little bit later this morning. So my name is Giampiero Cavalieri. I'm an investigator within the EpiPGX consortium. So EpiPGX was established to identify genetic biomarkers of treatment response for epilepsy, so pharmacogenomic markers for epilepsy. At the time that EpiPGX was being conceived, there were a number of different biomarker studies across Europe, and really EpiPGX was about consolidating those pharmacogenomic biomarker studies. And they were being consolidated in 2 different ways. First of all, by standardizing phenotyping across sites. So in other words, when a patient is phenotyped in Dublin, say, for epilepsy pharmacogenetics, we're following similar criteria to the phenotyping that's happening in Brussels. And then the second way was through consolidating and also generating additional genotype data on these patients. So the work packages, broadly speaking, are divided into two groups. The first set were around establishing infrastructure, so that could be criteria for phenotyping, a centralized database to store those phenotypes, a centralized database and infrastructure for storing the genetic data. And we're going to hear a talk later on from Roland Cruz around that infrastructure. And then the second broad group of work packages were around discovery, the discovery related to prognostic and predictive markers, say, for example, early response to, say, first antiepileptic treatment, broad refractoriness within epilepsy, or indeed a late response, such as that classic response to Keppler, so specific response to a particular antiepileptic drug after previous refractoriness. And we'll hear a talk from Holger Lerche later on that, and also Costin Liu on refractoriness and comorbidities. Then we had a work package on adverse drug reactions, so I'll present briefly on that later, and also on teratogenicity associated with sodium valproate. One of the key outputs of EpiPGX is this consolidated biobank, so we've phenotyped in the region of 10 to 12,000 people with epilepsy. These have been phenotyped across multiple different European sites. We have data outcomes on about 40,000 individual trials for epilepsy drugs. We have GWAS data on about 11,000 individuals and exome data on around 3,000 individuals. And a key feature here is that we're constantly updating these genetic data sets. As epilepsy samples are sequenced through other projects, the data is integrated into EpiPGX, so we're enriching the cohort constantly. So just to emphasize, a key output of EpiPGX is this centralized cohort. So phenotypes are entered into electronic case report form that's stored at the University of Luxembourg, and similarly that GWAS and exome data is in a centralized infrastructure that can be accessed securely by particular analysts. So in this way you get good quality processes around the processing and analysis of genetic data. So I hope you enjoy the EpiPGX talks that will happen throughout this morning and this afternoon. Thank you.
Thank you. So we move on to the EPISTOP project. So just your sec, please.
So thank you once again for the possibility to talk a little bit about the concept of EPISTOP project. Yeah, I manage. The title of the project is a long-term prospective study evaluating clinical and molecular biomarkers of epileptogenesis in a genetic model of epilepsy, tuberous sclerosis complex. Tuberous sclerosis is a very good model for analysis of epileptogenesis because up to 70% of patients develop epilepsy during the first 24 months of age, with very high incidence of autistic behaviors and intellectual disability. So it's a very good model for these genetic studies as well. And we are a group of 16 partners in the project. There are 5 laboratories across Europe and United States, and 10 clinical sites from different countries in Europe. And we have also one partner from Brisbane, from Australia. And there is one partner, administrative partner from Italy. We have 2 aims, because the project came from our observations from our clinical studies done in tuberous sclerosis patients when we observe a significant number of patients with severe forms of intellectual disability caused by epilepsy. So it's a very good example of epileptic encephalopathy. So the first aim was to decrease the number of patients with the intellectual disability in the group of patients with tuberous sclerosis. In 2011, we published our preliminary study, and EPISTOP was continuation in a larger form of this study. The first aim was to prove that this early treatment, because we identified a group of patients with prenatal diagnosis or very early diagnosis before 4 months of age of tuberous sclerosis. And in this group of patients, we analyzed EEG every 4 to 6 weeks to identify some EEG biomarkers of development of epilepsy in this group of patients. And we started to treat at this step before clinical seizures. So the first aim was just to prove that this preclinical, pre-seizure treatment may significantly decrease the number of intellectual disability in this patient. So it was the first aim of the study. The second aim was even more interesting because it was prospective possibility to analyze clinical and molecular biomarkers during the development of epileptogenesis until the age of 24 months of age. In order to carry out the study, we recruited to the project 101 newborns and infants before epilepsy, and we followed them up to 24 months, doing this EEG every 4 to 6 months in this period of time. We also had the possibility to collect in prospective way blood for different molecular analysis at the beginning, at enrollment to the study, at the beginning of abnormal EEG, at the clinical seizures, and in the 24 months of age. And with a significant number of different molecular studies, we are going to analyze different molecular biomarkers. And also we are analyzing brain MRI, which is these studies are also done several times during the follow-up. Yeah. We have 8 work packages. The most interesting work packages from clinical point of view, it's EEG. So we are going to identify the specific EEG biomarkers doing this, in fact, video EEG, 1 hour video EEG we are doing. So Lieven Lage is the leader of this work package, and he has a presentation today. We have also Floor Janssen, who is a leader of work package related to the neuroimaging studies, brain MRI, and different technologies related to brain MRI. And also Paolo Curatolo from Rome. He is a leader of the work package on neurodevelopment, on intellectual disability, on autistic behaviors. So we are going to analyze these biomarkers, and we are going to connect this with some molecular studies done in the work package 3 with the leader of David Kwiatkowski. He has also lectured today, and Eleonora Aronica, a leader of work package 5. So we are analyzing different biomarkers, clinical and molecular biomarkers in this group of patients. And we are very eager to get the results. We got the permission to finish the study 6 months later, so we are finishing in April next year. And hopefully we are going to invite also the leaders of other work packages to Warsaw next year. Thank you very much.
Thank you very much. And now we will have an overview of the EPITARGET project by Professor Merab Kokaya.
Good morning. It's a great pleasure to be here, and it's also a great excitement to finally arrive to this point where we have this meeting. And I would like to join David and thank everybody who participated in making this to happen. So shortly about EPITARGET. So it's targets and biomarkers of antiepileptogenesis. So mostly we concentrate on the process of epileptogenesis. This is a relatively big consortium. You can see here it's 9 countries, 18 groups. It's 5 years, and the starting date is December 2013. I think there is some mix-up with the slides. This was not... Anyway, so I will try to make best out of it. So here is the repetition of this slide. It was made in Keynote, and then something has happened So it's the same information here, anyway. So, okay. The major objectives of our project, as I mentioned, is targets and biomarkers of antiepileptogenesis, and there were three major objectives. The first of all, of course, to identify novel biomarkers and their combinations. So the combinatorial approach is a key in this project. Then we would like to address the mechanisms of epileptogenesis, and again in a combinatorial way. And this is because, as we all know, the epilepsy is a very multifactorial and complex disease. So the idea was that we have to look at the combination of the biomarkers and combination of the treatments to be able to effectively develop novel treatment strategies. And then, of course, we wanted to validate all the animal data that we obtained in Objective 1 and 2 in human tissue. In the Objective 3, and to try to correlate the animal data with the data that we can generate from the human material. Okay, it's not happening. Okay, so here is the overall design of the project, and you can see that the animal studies are designed in sort of coherence with what we can also validate in the human material. We mostly deal with the adult insult generated or post-insult epilepsy models. So there is an initial insult, mostly status epilepticus or TBI, and then there is the epileptogenesis process with a latent period. This is not really okay. And then the spontaneous seizures develop. And then we were trying to address different time points during this process of early and late epileptogenesis, and to match it with a human material that is available to validate the data obtained in animal studies. So according to the objectives, the work packages were divided accordingly. So the first work package concerns the biomarkers. Second and third work packages concerns the mechanisms of epileptogenesis and to develop the ways of delivering the treatment into the brain. And then the work package 4 is a validation in the human material. And we had, of course, dissemination and management work packages. Okay, I won't go into the details of the results because these will be presented during the course of the meeting today, and you will hear about the outcomes of the project. But overall, our productivity was about 114 papers that are already published. There are 7 more submitted in the pipeline. I'm glad to see that the most of the publications are in the high impact factor journals. As I mentioned, these results will be presented in specific presentations today. Some highlights that I wanted to bring from the project. There was a review paper published in Lancet Neurology concerning the biomarkers, and we were also the first to publish the common data elements and their management in the animals. And we are in the process of creating the animal database, which is perhaps one of the first, or if not the first. And we would like to share it with the community later on when it's in place, and it is based on the REDCap database. We also have the human database developing in the same sense. So I think this was my last one. Yes, so thank you very much, and enjoy the presentation.
Thank you. Thank you. So now we'll have an overview of epiXchange project from Michele Simonato.
Okay, thank you, and I also wish to join the others for thanking everyone that made this possible today and everyone that accepted to participate. I think this is a probably unique opportunity for the epilepsy community. And just briefly, I would like to offer you an overview of what was epiXchange. epiXchange was a YAP project that ended last year. YAP means industry and academia partnership and pathways, and that's thought as a instrument, a funding instrument by the EC, with the philosophy of promoting intersectorial research. epiXchange was probably the smallest of the projects that are presented today, and included only 2 academic partners and 2 industry partners. But the idea was to have a very strictly balanced contribution between the 2. So, in terms of manpower, exchange, and everything, everything was to be perfectly balanced. And the academic partners were from Italy and from Sweden, and we had 2 small medium enterprises, one from Denmark and one from France, that, as you can see here, over the 4 years of the project, exchanged tools, information, knowledge. We had a transfer of knowledge plan that included moving researchers from academia to industry, and not from industry to industry or academia to academia. And we also shared management, quality assurance, and regulatory issues, and so forth. The aim of epiXchange was to develop new unconventional strategies for the treatment of partial epilepsies, I should say, probably focal epilepsies now. And the essential idea was to deliver therapeutic proteins, and specifically inhibitory neuropeptides and neurotropic factors, directly in the epileptogenic area. And to do that, we developed 2 alternative strategies. One was based on a gene therapy approach, and we used this novel kind of vectors. These are HSV vectors that are called amplicons, in which essentially all the viral genome is substituted by a concatenator of therapeutic genes. And of course, these were injected directly in the epileptogenic area. And the other approach was a cell therapy approach based on encapsulated cell biodelivery systems. And you will hear more about these approaches later today. And especially this last one is now very close to being implemented in clinical application. As I said, the group was small. I think that the original epiXchange deserves some credit for what is happening between today and tomorrow, because in the last final meeting of the project of the consortium, we decided to take very seriously the recommendations of the EC to do dissemination and to do outreach activities. And for that, we decided to have a meeting that was called State of the Art and Opportunities for the Future in Epilepsy Research in the EU. We invited all other European projects that were directly involved into research in epilepsy. And as you can see, you can recognize the faces here that just came to this podium. And also we invited patients, organizations, and other stakeholders, including industries, charities, and funding agencies to discuss the possibilities to support epilepsy research in the future. So in a way, this initiative that we took at that time informed the initiative that we had taken in the last year, and that to this meeting today. Of course, we expect this meeting to be much more productive and to have an even greater impact for the future. So I wish you the best for these days and to enjoy the meeting. Thank you.
Thank you. Now we will hear from Helen Cross about EpiCare.
Thank you very much. And I'd like to also thank everybody who's been involved in the organisation and feel privileged to be here representing EpiCare, which, as you'll see, is slightly different so-called project to those that have been presented already. But really, we see ourselves as the clinical way forward and perhaps looking at the future from today with regard to clinical translation into clinical, of research findings into clinical practice. So EpiCare is a European reference network for rare and complex epilepsies. It is one of 24 European reference networks for rare diseases that were approved by the European Board of Member States in December 2016 and launched in Vilnius in March of last year. And it's one of 3 neurological European reference networks. We're there for rare and complex epilepsies, along with ERND for rare neurological disease, and also NMD, neuromuscular disease. We lobbied for our European reference network on the premise that increasingly, particularly the complex epilepsies, we're looking at a group of rare diseases for which there is a common symptom. And this because of the ways forward in imaging, genetics, other investigations, we're getting increased individual diagnoses, and perhaps we should be utilizing that knowledge to move towards a different way of thinking of treatment. We are a network of 28 centers across 13 countries. The premise for putting forward our network was to have 28 centers of specialist centers that have been approved by their individual country member state as being specialist centers, but also having the criteria of having the patient base within the rare disease cohort and also the knowledge base and utilization of specialist investigations to be considered specialist centers. But also recognizing that we had to have a limited number of centers for the recognition of the network, that moving forward there will be further calls later this year for other centers to perhaps join. And also there is a plan for those countries not represented, maybe not having centers of the appropriate expertise, or indeed not included in the original cohort, that will be associate centers. The way that this was set up was that initially we had to put forward our application, but this was not for initial funding. In parallel, we could apply for funding for coordination, and indeed 23 of the networks, including our own, were funded for 5 years by CHAFIA. But actually, the way we had to set it up, we set it up into 2 portions, diagnostic and therapeutic modules, trying to move forward particularly with the premise that we were going to use e-tools and virtual tools in order to move forward. Diagnostic modules looking at improved laboratory diagnostics, neuroimaging, neurophysiology, neuropsychology, and neuropathology, but also moving towards therapeutics, looking at targeted medical therapies. Surgery, epilepsy was one of 2 original pilot European reference networks led by Philippe Rivlin and myself, which continues as an arm of this, where they again had work packages for e-assessment, virtual assessment, aiding in assessment of individuals for epilepsy surgery, and also regular case discussion involving many centers utilizing a web-based tool. Then we have neonatal seizures and dietary therapies, but of course there are also the cross modules, not least looking at increasing registries in order to know where the patients are, guidelines, protocols, looking at management, diagnosis and management, education and training is key, And although this is essentially a clinical network, research and clinical trials are inevitably part of this and is seen to be integral. Further, the dissemination arm, in all parts of this, the patient groups are paramount. So there is patient participation in each of the modules, and also the electronic patient advisory group is also integral to this and part of our management board. Again, even though we launched in March last year, we had our first annual meeting in June last year. One slide is not enough to put up all our achievements over that initial period, but this just is there to emphasize that, yes, we're moving towards e-tools, namely looking at post-processing of MRI, an ongoing project from Epilepsy, but also new projects, computer-based systems for standardizing EEG reporting, and also for looking at neonatal EEG on a 24-hour basis, but also other aspects, namely the continued epilepsy surgery case discussions to aid local management, but also moving towards utilization of the EU centralized collaborative patient management, clinical patient management system to also discuss non-surgical cases. Registries are key. Where are the patients to indeed roll out perhaps possibly new therapies, new treatments, looking at cohort studies, and that is in development. And of course, to be here as part of discussion of the future, as part of epiXchange, is also a key achievement, and we're very grateful for that. Thank you very much. What we want to do is move forward to moving our basic science into patient care. Thank you.